afternoon, everyone. My name is Dr. Diallo Kanny. I'm a research associate at Centre Suisse de recherches scientifiques in Cote D'Ivoire. And today I'm going to take you through lab experiments for DNA extraction. So as you may all know, DNA extractions is a really important step before doing any molecular biology work. And today I'm going to present two different methods for DNA extraction. The first one is a crude DNA extraction that can be done very quickly and simply, but has limitation in the downstream applications. And the second one is a more advanced method using kits. So you can use any kit you want, but we are going to use the Promega Wizard DNA extraction, genomic DNA extraction kit. So I welcome you into our lab at Centre Suisse. So this is our one of the molecular biology lab that we have at Centre Suisse. And I have prepared some of the materials that we are going to need to do our experiment today. So we are going to do DNA extractions from bacteria that have I have cultured since yesterday. So the first thing that we are going to need is our bacteria. And I've cultured some Moraxella on blood agar. So I'm going to show you under the hood. Depending on the organism you're using, you may or may not need to work under a hood. Because if they are dangerous or pathogenic organism, it's safer to work under the hood. Otherwise, you can work on the bench. So we are going to use the plates. We need some tubes. So here I've prepared a tube with PBS for the DNA extraction, the crude DNA extraction, or boil it. I prepared as well a floater to incubate in the water bath. And I've just going to use some aluminum foil to make sure that we seal our tube very well so that it doesn't pop when it's boiling. The other thing that we are going to use is some loops to take the bacteria from the plate into the PBS. And that's pretty much it for the first experiment. So now we're going to start. So I'm going to take my bacteria and my tube under the hood. I'm going to take my loops, and that's it. And I'm going to take a little bit of aluminum foil. Don't need a lot. Okay, so I have put 1 ml of PBS into my Eppendorf tube. Now I'm gonna take my loop and take some bacteria. You can see the bacteria growth on the plate here. For boilate, you do want to take quite a lot of bacteria. So you can take as much as possible. You want to see a visible lump. All right. And then you're gonna resuspend this in the PBS. You can see the PBS becoming turbid. The bacteria is going into the solution. There you go. You want to make sure you avoid any aerosols that can contaminate your surface, especially if you're not working under the hood. I'm going to throw this. Now, I'm going to seal my tube with the aluminum foil. You can use a tube stopper, or there are many different device you can use to stop this from popping out, but just as an inexpensive tool, we use aluminum foil. Then I will take this to the water bath for incubation for 5 minutes. We are now going into another section of the lab where we're going to incubate our bacteria in the water bath. We are here in our main molecular biology lab. At 95 degrees, so that we can incubate, boil our bacteria solution. Let's see here. I'm going to put this here. And then we're gonna set a timer for 5 minutes. So we're going to come back in 5 minutes and continue with the process. Now the incubation is done for our crude DNA extraction. So I'm going to take out my tube. And you can see it has boiled. I will just wipe it out of it with some paper. I'm going to remove the aluminum foil. So what has happened here is that the cell, as they are boiling, are going to break the different nucleus where the DNA is. So now the DNA has been released into the liquid, into the PBS. And so all you need to do is to centrifuge your tube to get the cells down, and the nucleus will be in your supernatant. So to do that, we use this machine, which allows us to spin down the tubes. So we are going to spin at 13 G for 3 minutes. To use this machine, you need to have an equivalent tube to balance. It's very important that you balance your tube when you're spinning. So I'm going to put the one tube here that we're interested in, and a balance on the other side to make sure that it's equilibrated when you're centrifuging. Close this. And I'm gonna run, like I said, for 30 minutes, at 13,000 G for 3 minutes. So you see, you have a pellet here with all the cells, and now the DNA has been released into the supernatant. My balance. DNA extraction, we can now transfer the supernatant into a clean tube, and this is the DNA that you can use for PCR and even for some Sanger sequencing. I've used it before and it worked well for Sanger sequencing as well. So to do that, I'll just again take a pipette, so you want to avoid touching the pellet. You want to only take the liquid. Okay, so now I'm going to label again. I'll put the name of the bacteria and what it is. So I'll put boilets, which means DNA extracted by boiling, and I'll put the date. So we are done for the DNA extraction. It's very quick and very simple way of extracting DNA. The second method that we are going to use is a more advanced DNA extraction using a kit. So we are using this kit, which is a Wizard Genomic DNA Purification Kit that comes with the reagents that are needed. So for this, the first principle is the same. You want to take some bacteria and put it in a lysis buffer. I will label with the name, what exactly it is, and then the date. For this, we are going to use the nucleolysis solution, which comes in the kit. And I'm going to take 600 microliter. Then I will take this time a 1 microliter loop, and again I will take some bacteria from my plate. This is enough. I will resuspend the bacteria. It helps to put it on the wall, because it's easier to resuspend, and then go up and down until the solution becomes turbid again. Bacteria is well resuspended. And again, the same way as we did for the first one, we're going to incubate for 5 minutes. It is important to note that the procedure will vary depending on the bacteria that you're going to be using. I'm using a gram-negative bacteria, so we can boil without any enzyme. But for some bacteria like Streptococcus pneumoniae, which are gram-positive, the cell wall are stronger. And so the boiling may not be enough to break the cells and then open the nuclei for the DNA to be released. So for those bacteria, you can use an enzyme that you add before boiling. It can be protein. The most common one is proteinase K, but you can use other enzyme depending on the bacteria that you're using. So for us today, we're using a gram-negative, so you can boil directly. Now we are done with the boiling of the bacteria in the nucleolysis solution from the Wizard kit. So I'm just going to remove the aluminum foil. Okay. So now I'm going to use a micropipette to add an enzyme. This enzyme is RNase A solution. The enzyme is going to degrade any RNA that you may have in your boilet. So basically, at this step, you want to get rid of any RNA in your sample so that you can extract only DNA, because you're only interested in DNA. This enzyme works at 37 degrees. So what we're going to do is add 3 microliter. The tube, you have to make sure that it has come back to room temperature, because if it's still very hot, it will denature the enzyme and it will not work. So you really want to make sure that your tube is back at the normal temperature, room temperature. You see the RNAs? You take 3 microliter of the RNAs, it's very small, and you add it to the tube that you had boiled. Okay. So you just invert a bit, and then we are going to incubate this at 37. So there are many options if you want to incubate. The easiest one is in a water bath. But here we also have a bead bath that keep at 37. So today I'm going to incubate here at 37 for 30 minutes. After 30 minutes, we come and continue with the process. Okay. So now we are back after 30 minutes of incubation at 37. So I'm gonna take my tube out, and then I am going to bring it back into the hood. Now, the procedure says that you want to let the tube come back at room temperature. So you let it come back to room temperature. And the next step is to add this solution from the kit. The name of the solution is protein precipitation solution. So what it's going to do is it's going to bind to all the proteins in this tube and then pull them down so that you can separate the protein that you don't want from the nucleic acid, which is the DNA that you're interested in. So we are going to add 200 microliter of this tube, of this solution. So I'm going to get 200 microliter. Add it to my tube. now that I've added the protein precipitation solution, I am going to vortex for 10 seconds, 20 seconds. But it's very important, this step, you have to really vortex strongly. Okay. And after that, we incubate at 4 degrees. So at 4 degrees, it can be on ice, like this. But you can also incubate in the fridge if you don't have ice buckets or ice in your lab. There you go. So just put it on ice, and we're going to wait 5 minutes. While we are waiting, we are going to prepare the next step. For the next step, we are going to need some isopropanol. So I've just poured some isopropanol from the bottle here. So now I'm going to label a new tube with the same label. Okay. And I'm going to put 600 microliter of isopropanol in this tube. The isopropanol is to pull down the DNA, to precipitate the DNA. So once we get back our sample with our bacteria, we're gonna pull down the DNA using the isopropanol in the next step. Our incubation is over at 4 degrees. So now we're gonna centrifuge at at 13,000 G for 3 minutes. As you can see, we have a very nice precipitate at the bottom of the tube, and your DNA is in this solution. So we're going back into the woods. Now we are going to transfer the supernatant into our tube containing isopropanol, the alcohol. So you want to avoid completely the pellet. It's better to leave a little bit of it than touching the pellet. So you add this to the ethanol, the isopropanol, sorry. And that's the exciting part, because you now start shaking gently. You should see some DNA. Okay. Now that you have mixed, we're going to go back and centrifuge for 2 minutes, and then you should see your DNA. So we are going to centrifuge now for 2 minutes at the same speed. So here you can see a small dot. Okay. So now isopropanol from our tube. You can see a small DNA at the bottom there. So what I do is I empty the ethanol, the isopropanol, and then I blot it onto a clean sheet of paper. Enough to remove all of the isopropanol. You can see your DNA at the bottom here. Then let me check. Yes, I will add 600 microliter now of ethanol, 70% ethanol. So you prepare 70% ethanol, 600 microliter of 70% ethanol. You can see it's very difficult for you to see, but there is some DNA floating in this tube. So you just rinse it out, like mix it a bit like this, and then you repeat the centrifugation for 2 minutes. After the centrifugation, you're again going to empty the ethanol, and then you're going to blot it. Make sure that you can see your DNA at the bottom of the tube. Make sure not to lose your DNA as you do this. As you can see here, it has moved up a little bit. Then we are going to let it dry, open, for 15 minutes. So now that our pellet is dry, the ethanol has evaporated completely, we can add our DNA rehydration solution. We're going to add 100 microliter of DNA rehydration. You can see the DNA is not yet in solution, so it is recommended by this protocol to incubate it at 65 for 1 hour. So I'm putting it at 65 for 1 hour. Now that we have incubated at 65 for 1 hour, our DNA is in solution, is dissolved in the solution. We can store our DNA at -20 or at 4 degrees for direct applications. The next step after you're done with your DNA extraction is to check the quality of your DNA, and there's many different approaches to do this.